All right, welcome to the Chapter 1 video. This is covering the introduction to microbiology history and how the subject came about, and we'll get into microscopy. So one of the first questions I always ask my students in the first day of class is, why do I need to study microbiology? And the majority of my students are going into nursing or dental hygiene, some sort of healthcare field. So take a minute and think about why you might need to study this topic. And in order to maybe fully answer that, you have to consider, well, what is microbiology? So microbiology is the study of organisms that are generally too small to be seen by the unaided eye. So we'll be looking briefly at some eukaryotic organisms such as algae and yeast, amoebas, some different worms. But we're going to focus this class on bacteria that are microscopic. You can't see them without a microscope. So, like I said, generally this topic is looking at organisms too small to be seen by the unaided eye. And a lot of misconceptions about these bacteria, or germs as people often refer to them. And 99% of bacteria are actually harmless, with a few or more than a few being actually beneficial. And less than 1% of bacteria are harmful or disease-causing, which, if you were thinking about why you need to study this topic going into healthcare, you'll be dealing with people that are sick caused by bacteria, viruses, and things like that. So this term flora was once used to describe bacteria, since bacteria were once classified as plants, and the term flora has now been replaced by microbiota because bacteria are in their own domain. They're not related closely to plants at all. So we refer to the microbes present in and on the body, human body, referred to as normal microbiota. Normal microbiota serve as a first line of defense against pathogens, causing preventative exclusion. So they take up space and nutrients, and prevent the growth of actual pathogens. The abundance of these normal microbiota on the skin, there's about 2 million microbes per square inch of your skin. In feces, there's about 100 billion bacteria per gram. And looking elsewhere in the soil, there's about 10 million microbes per gram, depending on the soil. And in the air that you breathe, 50 to 100 microbes per cubic foot. So these bacteria, microbes, are all over the place. In humans, inside and on the body, we are consider composed of about 100 trillion cells. Only 10% of those are actually your cells. So that means there's 90 trillion microbial cells. That includes bacteria, fungus, maybe some protozoa in some cases. 90 trillion microbes help make up your actual, are found coded in and on your body. Developing those normal microbiota, basically, we develop those after the amniotic sac breaks during the birthing process. So inside the amniotic sac, the environment there should be sterile. So once you come into contact with the outside world and be handled and eat food and things like that, you develop those normal microbiota. There's many germ-free areas of the body. Those are listed in your book in Chapter 1. But inside, muscle tissue and organs, those would tend to be sterile areas of the body. Now, some normal microbiota can actually serve as opportunistic pathogens. Opportunistic pathogens are organisms that normally do not cause disease, but given the opportunity, meaning that they gain entry into part of the body that they're not supposed to, or you're immunocompromised, they can cause disease. Some examples of normal or some opportunistic pathogens would be organisms like your Staphylococcus aureus, which you may have heard of, more commonly referred to as MRSA, methicillin-resistant Staphylococcus aureus, But the organism Staphylococcus aureus is found on pretty much everybody's body, and it's an opportunistic organism or pathogen. So a pathogen is a little bit different than an opportunistic. A pathogen can cause disease in a healthy individual. So whereas opportunistic, you either have to be immunocompromised or it enters into the body where it's not supposed to. A pathogen can cause disease in a healthy individual. And what we're looking at with pathogens, we're looking at infectious diseases. So when a pathogen overcomes the host immunity or resistance, disease results, and there's changes to normal body function. So these are different than other diseases because an organism is causing an infectious disease, whereas a disease like most cancers are not caused by an actual disease or an autoimmune disease. It would be more genetic factors. When classifying diseases, we can classify them as emerging disease, reemerging diseases. But emerging disease is something new that has not been seen historically. So your MRSA, AIDS, E. coli, 157H7 would be emerging diseases. MRSA first seen basically in the 1980s, and now in the 1990s, there's actually some resistant strains of vancomycin-resistant MRSA as well, which vancomycin was considered the drug of last resort. So MRSA is an emerging disease because it's not seen, relatively speaking, historically, first popping up in the 1980s. E. coli 157H7, this is a toxin-producing strain of E. coli. Now, E. coli, everybody has E. coli inside of their colon. But 0157H7, this is a strain of E. coli that is disease-causing, first seen in 1982. So it's a leading cause of diarrhea, bloody diarrhea worldwide. And then AIDS, or acquired immunodeficiency syndrome, caused by the HIV virus, first identified in 1981. It would have been in the population much longer than 1981, but wasn't really known until 1981. But this is an emerging disease, sexually transmitted disease affecting both males and females. And then reemerging disease, this is a resurgence of old diseases due to extensive travel, lax attitude towards vaccination, and increased population with immunocompromise. But most of the time, we're dealing with a lax attitude towards vaccination. So either parents not vaccinating their children or actually adults not getting booster shots as well. So those diseases, a couple examples would be tuberculosis, pertussis, measles would be some reemerging diseases. So tuberculosis is considered a reemerging disease, increasing at a rate about 2% each year. Bordetella pertussis. This is a preventable disease through vaccination, but it's kind of endemic in the United States, and we actually have increasing numbers because people are not vaccinating. Looking at some microbes in the environment and their roles in the environment of decomposition, this is a field referred to more or less as biodegradation. So microbes are used to treat sewage, break down toxic waste products, so in oil spills and other pollutant examples. Decomposition plays a role in food spoilage, so bread, mold, soured milk, things like that, resulting in billions of pounds of food lost each year due to microbes. Now, bacteria play, with that decomposition, play an important role in element cycling. So the carbon, nitrogen, sulfur, phosphorus cycles are relied upon by bacteria and other microbes. So either through carbon fixation processes of photosynthesis or decomposition, which is recycling of those elements back into the environment. Crucial role played by microorganisms. One of the main goals of the first unit is to, get people to understand, and not to become germaphobes, but to understand that how crucial these microbes are in our everyday lives. These microbes are also involved in food production, such as alcoholic beverages, cheese, yogurt, bread, fermented foods such as sauerkraut and vinegar. And other microbes as food would be mushrooms, which are a type of fungus. So looking at some advantages of studying microbiology, we can get fast results and grow these things by the billions of cells per milliliter, and have fast results within a day or two, or even hours, depending on the type of procedure that we're actually being performed. Microorganisms display the same fundamental and genetic properties as found in higher life forms, so they can be used as model organisms for growth. And the space requirements are usually minimal, so we don't have to have large facilities such as a zoo for large animals to study. And relatively inexpensive, so we're using simple media with sugars and nitrogen sources to grow these microbes. All right, now switching gears to a history of microbiology, basically looking at how the field came about. Early scientists or people were looking for answers to basic biological questions. So, is spontaneous generation of microbial life possible? What causes disease, and how can we prevent disease and infections? So, a couple of the major events that led to the field of microbiology would be the debate over spontaneous generation, and which is really started by Francisco Redi in 1668. The dates here are not important, but you should be familiar with who these people or what these people contributed. Another crucial event would be the invention of the microscope, so by Anton von Leeuwenhoek in 1673. So, two events that helped lead to the development of the field of microbiology. So, we'll look a little bit more closely at this. So, looking at the debate over spontaneous generation, basically, this theory has been around for a very long time. Proposed by Aristotle in the 300 BCs, at some point during his lifetime. And spontaneous generation is a hypothesis that living organisms arise from non-living matter, and a vital life force helps form the life. And the conflicting theory would be biogenesis, which is the hypothesis that living organisms arise from pre-existing life. So Francisco Redi, he begins this looking at the spontaneous generation. He's interested in the spontaneous generation of maggots, and he basically does a couple of experiments. First, he takes a flask that's unsealed here and with a piece of meat. He takes a flask with a cork in it, and seals that from the outside environment. And what he observes is, well, maggots form on that piece of meat, and then flies emerge. Here, with the flask sealed, no maggots, no flies. So what he observes is that, well, flies are buzzing around this meat, and he proposes that these flies are laying an egg, and those eggs develop into the maggot, and those maggots develop into the fly. The critics of this experiment would have been that, well, that vital life force needs air in order to survive. So what he does then is he puts a cheesecloth mesh over the top of a third flask and does this experiment, and he sees flies buzzing around, and he then notices maggots forming on top of this cheesecloth, but those maggots quickly die because they don't have a food source. So right here he's giving evidence for the theory of biogenesis, that life from the fly is needed for life of the maggot. Anton von Leeuwenhoek, a few years after Redi does his simple experiments, develops the first microscope. So he's commonly referred to as the father of microbiology, and discovers that microbial world in 1673. Before that, we didn't really have any idea that, in a drop of pond water there are organisms swimming around in there. So he's the first one to visualize these. Looking further at the debate over spontaneous generation, John Needham in 1745, what he does is he is working with microbes. He puts boiled nutrient broth, much like what we use in the classroom, and he puts it into a covered flask. So this would be nutrient broth with turbid microbial growth, turbidity meaning that there's microbes present. So what he does is he heats the broth, then places it into a flask and seals it. So boiling the flask is thought to kill all the life, but what he sees is turbid microbial growth. So this is actually providing evidence for spontaneous generation because he sees that microbial growth. Lazzaro Spallanzani in 1765 looks at Needham's study, and he suggests that, well, microbes from the air had entered this flask during that time that he transferred the boiled broth into the flask. So what Spallanzani does is he boils the nutrient solution and then seals the flask immediately. So what he sees is no growth, so it's clear, indicating, he killed the life in that and gives support for biogenesis. Well, the critics of this said, well, again, air is needed for life, so this debate continues. In 1861, so backing up, 1765 to 1861, almost 100 years later, Louis Pasteur demonstrates that microbes are present in the air, and he debunks the theory of spontaneous generation once and for all. And what he does is he develops the swan neck flask. So this flask here that he develops, he can put nutrient broth in here and then boil it. And what happens is the steam can escape, and any microbes that are in the air can fall into this little neck of the flask here. But this broth remains sterile for long periods of time. Unless you tip this and introduce that media to that contaminated area and then reintroduce those organisms by tipping it back, this broth will stay sterile indefinitely. So the spontaneous generation debate led to the scientific method. So observations leading to questions, and the questions generating hypotheses, and the hypotheses are tested through experiments. So not only did the debate over spontaneous generation lead to the field of microbiology, but it helped form the scientific method. The second question that scientists were looking to answer was what causes disease. So Louis Pasteur developed the germ theory of disease, meaning that microorganisms cause disease. And then Robert Koch in the 1800s, early 1900s, studied causative agents. So a causative agent would be something that directly causes a specific disease. And he demonstrated anthrax was caused by Bacillus anthracis. In order to do this, he developed postulates, so steps one must take in order to prove a microbe causes a specific disease. The first one is the suspected microbe must be found in every case of the disease and absent from healthy hosts. The suspected, meaning like if he's looking at anthrax, for example, any person or animal with anthrax must have Bacillus anthracis, and anybody who's healthy does not have Bacillus anthracis. And then we must be able to take that suspected microbe and grow it outside of the host, so in the laboratory. So we take that sick individual, we isolate Bacillus anthracis from them and grow it in the lab. When that microbe is introduced into a healthy person, that new person must acquire the disease. So if we take that from the lab and put it into you who are healthy, you must then get anthrax. And then we must then isolate that organism from you again and grow it into the lab. So re-isolate it. So in order to prove a causative agent, it needs to follow these postulates. Now, Koch's work led to vaccine development, which led to the eradication of smallpox, which smallpox is the only virus that has ever been eradicated from the planet. And we have a significant decline in polio, measles, other childhood diseases, and knowing what causes a disease helps lead to the development of antibiotics as well. And then finally, how can we prevent disease? So there's a couple people here, Ignaz Semmelweis and hand washing. Ignaz Semmelweis, he's the first person to ever show a direct relationship in hand washing and a drop in disease. So he was a medical doctor, and he hypothesized that his medical students were transferring what he referred to as cadaver particles from autopsies into delivering rooms. So mothers would develop this puerperal fever or childbed fever, and it was known to be caused by a bacterium in the genus Streptococcus. So he began requiring medical students to wash their hands with this chlorinated water, and in 1 year he sees a drop from in mortality rate from 18.3% from mothers who would get this disease to 1.3%. So just by hand washing alone, we have a significant decline in disease. He was unsuccessful in gaining support for his method of hand washing because when he took this information and presented it at medical conferences, he was basically shunned because doctors had this mentality that there's no way that they were the reason why these mothers were getting sick. So he later developed depression, was committed to a mental hospital, and there was basically reports that he got into some altercation which led to an infection, and he ended up dying from an infection caused by an organism from the Streptococcus genus. So this organism that he was looking to prevent ended up killing him in the end. And then how can we prevent disease? Another instrumental person, Joseph Lister, he's stated to have saved more lives in the 19th centuries than all the wars during that time had sacrificed, because he introduced his system to prevent surgical wound infections, referred to as aseptic surgical techniques. We will use aseptic techniques in the lab in order to prevent organisms from the environment or yourself into our experiments. He used methods to prevent contamination of unwanted microorganisms onto surgical tools and things like that. During the Civil War, a field surgeon may have taken a scalpel and wiped it off on the sole of his boot before moving on to the next patient. So there was no sterilization or even disinfection procedures that were being followed until Joseph Lister began looking at these new techniques. The last portion of this chapter is microscopy. Microscopy using microscopes, lenses to magnify images. And there's three quality factors that determine the quality of an optical image. There's magnification, resolution, and contrast. Magnification, we will use a compound microscope in the lab, which is a two-lens system. We have an objective lens in the nose piece, and we have an ocular lens in the eyepiece. So our total magnification is always the magnification of the objective lens times the magnification of the ocular lens. So our objective lenses are 10 times magnification. Our ocular lenses are 10 times magnification. Our objective lenses are either 4, 10, 40, or 100 times magnification. So basically what that means is the most we can magnify in the lab is 1,000 times magnification. So what was happening is that the glass is actually bending the light, and we will see a larger image through our oculars. But what happens is the image is actually inverted and reversed and enlarged at the same time. So we'll do some experiments in the lab to actually show how the microscope is actually reversing the image. Resolution is the ability to separate two objects that are close together so that they're seen as two objects. So basically resolution is the focus, more or less. So being able to clearly define, the borders of a cell and things like that, and separate them and distinguish them from one another. And resolving images is one of the things people struggle with in the lab. One of the ways that we will help our resolution when we're at high power, so when we're trying to view our specimens at 1,000 times magnification, is we use oil immersion. So oil immersion basically prevents the light from refracting or bending away from our objective. So it helps focus the light through the sample. We'll talk more about that in the lab. And then the contrast is differences in intensity between two objects or between an object and the background. So most microbes are actually colorless, so they're hard to see um, against a background, a colorless background. So we use stain to provide contrast. We will do a simple stain procedure, which will just stain cells with a simple color, or we will use differential stains to differentiate organisms from one another. There's different types of microscopy. There's all different types. We use light microscopes, so it's called bright field or light microscopy. And this is one of the most important tools for studying living organisms. So we can view them up to 1,000 times magnification. Other electron microscopy practices can magnify in excess of 100,000 times, but these organisms are dead at that time. So electron microscopes, we can see very fine details of cell surfaces, internal and external structures. Here's a look at, this is not the same brand as ours, but more or less the same setup. We have the ocular lenses. These are 10 times magnification. The objective lenses here, these would either be 4, 10, 40, or 1,000 times. There's the specimen stage. There's a light condenser underneath. This is an important tool under here because this is what will actually increase or decrease the amount of light going through the specimen into the objective. We'll have the light source on the bottom, and then there will be a rheostat. Ours is actually over on the side. So looking at the types of microscopy, the electron microscopes, there's transmission electron microscopes, which is high resolution. We can see internal structures of the cells, and scanning electron microscopes, which are surface details, things like that. This is a transmission electron microscope image. This is 1 micrometer, and you can see the nice details inside that cell. And the size of these things compared to a simple light microscope is quite drastic. These would be scanning electron microscope images. These colors that you see would be digitally enhanced. For the most part, a lot of this stuff is not this brightly colored. So looking at some staining techniques, we're going to use staining to provide contrast and resolution between the microbe and the background, and it helps make the microbe more visible. Like I said, there's simple stains, which is just one stain or dye used to provide contrast. We will do this with like a cheek cell stain, look at cheek cells using methylene blue. And then there's differential stains, which is 2 or more stains and dyes to provide contrast. And then there's some special stains that actually stain certain cell structures or backgrounds. So simple stain, again, using a single stain to stain cells. Some different stains that would be methylene blue, crystal violet, safranin. We'll use all of those in the lab. Differential stains. There's the gram stain. That's the most important for our class here. We will be doing this quite often. Acid, fast, and endospore stains, we're not going to be doing those, but we'll briefly talk about those. The gram stain provides differentiation between what are referred to as gram-positive and gram-negative bacteria. Gram-positive bacteria stain purple as a result of this procedure, and then the gram-negative stain pink. So with the gram stain, we start with crystal violet on the slides. After about a minute, we rinse those slides and apply Graham's iodine, which is a mordant, and it binds to the crystal violet and makes the crystal violet a large molecule difficult to wash out of the cell. We then use alcohol to decolorize the crystal violet, and then safranin is used as a counterstain. So we'll talk about how this procedure actually works, but it's a 4-step process resulting in either pink or purple cells, and it has to deal with the cell wall structure of these cells. So we'll talk more about that in a later chapter. There's the acid-fast stain, which is used to stain organisms from the genus Mycobacterium. Acid-fast bacteria are bacteria that contain mycolic acid in the cell wall, which is a waxy substance, and it basically resists certain stains. So it's used to differentiate between acid-fast bacteria, which are organisms with mycolic acid, or non-acid-fast bacteria, which do not have mycolic acid. So this is an important staining procedure for organisms such as Mycobacterium tuberculosis. Causative agent, tuberculosis. Endospore stain. This is used to stain cells which form endospores, and those that do not form endospores. An endospore, we'll talk about later in chapter 3, is a protective, dormant structure formed by some gram-positive organisms from the genus Bacillus and Clostridium, to name 2 clinically significant genera. But you can see here the green endospores. This is the protective structure, and the cell would actually be dormant at this time, and they are not metabolizing at all during the endospore state. These cells over here are not in the endospore state at this time. They are in the vegetative cell state. Okay, so vegetative meaning that they are actively metabolizing at that stage. Then there's the capsule stain. A capsule is a structure that some bacteria form. It's kind of it's a polysaccharide secretion. We'll talk more about capsules, but here you're actually staining the background of the specimen, and the capsule is not. It's resisting the stain. And then the last type of stain would be the Flagella stain, used to stain the flagella, which some bacteria produce, and it's normally too thin to be seen just with the normal microscope. Okay, so to end out this chapter, there will be the Chapter 1 quiz on e-learning. Before you complete the quiz, I would fill out the study guide and study from that study guide before completing the Chapter 1 quiz. The microscopy portion of this chapter will be in the Chapter 2 quiz, and you'll see that when you click on that module. It'll say Chapter 2 with microscopy. So just be prepared for that slight difference there. Now, the Chapter 1 study guide is in your course pack. The Chapter 1 study guide is also updated, available on e-learning. So we talk more about that when we meet.